In chapter 4, we are going to talk about the archaeal cell structure and how they differ from the bacterial cell structure that we have seen in chapter 3, and what are some of the similarities as well as some of the dissimilarities, as well as how they may have some of the characteristics of the eukaryotic structure. So here, what we are looking at, that archaea and bacteria were jointly referred to as prokaryotes historically. But today, they are recognized as a distinct taxa, like an entirely different domain with a distinguishing characteristic. And thanks to the classification system or the molecular classification system based on the 16S small subunit ribosomal sequencing that was done by Carl Woese and Fox, George Fox, and that is what is recognized now. They are highly diverse with respect to morphology, physiology, as well as reproduction and ecology as well. The archaea are essentially known for growth in anaerobic condition in hypersaline, which is also known as the halophilic environment, very high salt, pH extreme, and high temperature habitats. And no archaea is till date is known to cause any kind of pathogenic damage or infection, because human body is usually not a proper environment for any archaea to grow. So here we are comparing the bacterial and the archaeal cell. So let's start with the plasma membrane lipids. We are going to see that there will be some different kind of bonding and structure. We are going to go into detail that mainly there are ester-linked phospholipids, whereas we are going to see that there will be ether-linked phospholipids that are present, and that could be diethers or tetraethers. We are going to see that sometimes that gives rise to a monolayer membrane, a membrane with just a single layer in some of these structures. The cell wall constituents are always peptidoglycan, present in nearly all cells, and some lack cell wall, just like mycoplasma we talked about. Whereas in the archaea, the cellular envelope is very, very diverse. And outside the plasma membrane, usually the most commonly they have an S layer, and this S layer may have another protein layer at the top, or may have a carbohydrate layer at the bottom, or some of them actually have a dual membrane, or like a diderm, that the term we introduced in the last chapter. And the peptidoglycan is usually always absent, but a similar structure known as a pseudopeptidoglycan or pseudomurine with a different with a different sugar and a different bonding and different type of amino acids instead of D amino acid. Some of the L amino acids are present in these structures, which is found in some of the archaea. Inclusions are usually always present, and the ribosomes are of the 70S size, but the proteins actually are very diverse, and the sequences are also diverse. But size-wise, the bacterial as well as archaeal ribosomes both are 70S ribosome. They contain circular double-stranded DNA, just similar to the bacteria, and the circular double-stranded DNA plasmid as well as usually found. We found some of the linear double-stranded DNA plasmids as well in some of the bacteria, which are usually not found in the archaea. And the capsule or the slime layer, which are very common to bacteria, are usually rare as well. And fimbriae and pili are very common in bacteria, and pili is common, and there is also another structure which is known as the archaela, which is a kind of a hybrid in between the pili as well as the flagella. And the archaea's architecture is very different from the flagella that is present in the bacteria. We're going to talk about it. And the motility structure is, of course, the flagellum for the bacteria and archaeolum for the archaea. And there are several types of multiple homologous tubulin and actin proteins that were found. We talked about those. Some of these multiple homologs of tubulin or actin are also found in, which are similar to the homologs of eukaryotic cytoskeletal, which are found in the archaea as well. And both can form some of these extracellular vesicles. So let's talk about the archaeal shape and arrangement. The Cocchian rods are actually common. Other shapes exist, but there is no spirochete-like or mycelial forms known yet. And unique branch and flat shapes have also been observed, and the sizes also vary. Usually the rods are about 1 to micron wide, 1 to 5 micron in diameter, micron in length, Cocchi 1 to 3 micron in diameter, and even 0.2 micron in diameter, some of the smallest one, which are symbiotic, lives along with another host, have been observed, we are going to see. And so far, the multicellular form can reach up to the 30 millimeter in length. These are some of the extremes of some of these archaeal sizes. So here we are looking at some of the archaeal cell and morphology. So here we are looking at a coccus which forms clusters. This is a Methanosarcina mezi. So you can see that they are very similar looking from the bacteria, although they are different, but very similar looking. On the other hand, this is a branched archaeal cell, Thermoproteus tenax, you can easily see. So here what we are looking at, that this was discovered in 2015 and it was published in 2019. And this is a new symbiotic nanoarchaeot. The similars of these structures are found elsewhere, but this is essentially a novel strain that was actually found. And so this one is the Candidatus nanoclepta minutus, this one, the small one. Whereas its host, which is the bigger one here, right here, the Z, that is the Zytosphaera tichirensis, and see that is the genus novel, species novel. So if you'd like to take a look at the paper, you can follow this link to look at the paper, that how it was discovered. So this was discovered from a geothermal area known as Tikitiare. This is known as Hell's Gate in popularly. And this is a small town of Rotoruas in the North Island of New Zealand in the geothermal area. And you can easily see that this is very similar to an environment that what you find in the Yellowstone in the United States, from where a lot of different types of archaea have been found. And they have collected that, and they have done extensive amount of metagenomic analysis, and you can just read the paper that how they actually characterize that. And there are several different forms of these type of bacteria with different, they morphologically similar, but with different sequences have been also reported from all different parts of the world. There are, as well as from the Yellowstone, some of these hot springs, as well as there are some marine forms as well. So these are some of the smallest one you can easily see, nanoarchaeot. On the other hand, here you can see that this is a filamentous archaea, and the archeon you can almost can't see except at the very top. So it's filamentous, and there is a bacterial biofilm. So all on the top, that what you are seeing, these are the bacterial cells which are adhering on top of the archeon, and they live in a symbiotic relationship with help each other. So these are some of the diverse cell morphology and sizes of archaea. So let's first then take a look at the archaeal cell envelope, and of course they are structurally very diverse. The bacterial cell envelope in the molecular makeup and the organization is very different from the archaea. So the S layer may be the only component outside the plasma membrane, which is similar to the bacteria, and some lack a cell wall. And slime layers are observed to mediate cell-to-cell interactions, but little is known about the composition regulation and is rare. So the archaeal membrane, which essentially encloses the protoplast or the cytoplasm, is composed of very unique lipids. So if we just look at the phospholipid, if you just look at the general structure of the phospholipid, you have a phosphate head group, which is polar, hydrophilic end, because it's polar, because you see that you PO4 minus, so PO4 minus, and you have a negative charge right here. And then right here is the glycerol linkage. And then you have two fatty acid tails, which are connected with the glycerol in here in archaea with the ether linkage. You can see that CH2OCH2, this through the linkage through the oxygen is essentially, this is the ether linkage. So this is different from the bacteria, where the hydrocarbons are attached with the glycerol in by ether linkages rather than the ester linkages, which is found essentially in the bacteria. The bacteria, this linkage is an ester linkage. We're going to see that. And the hydrocarbons are essentially derived from the isoprene unit, which is a 5-carbon branched molecule. So you can easily see that you have this 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and these are the isoprene unit, which can be branched. And this is not found in the bacterial or the eukaryotic plasma membrane either. Sometimes what happens, the ends of the phospholipid bilayers. So here you can see that these are some of the integral proteins going across the plasma membrane. And here you can see that this is the outer leaflet with the 2 phospholipid tails. Here, this is the inner leaflet with the 2 phospholipid tails. But sometimes in some of the archaea, and this is a bilayer of what is called the C20 diethers. That means the carbon chain length is C20. Now here you have a C20 and C20. Sometimes they are covalently joined to form one end to the end, C40, 20 plus 20, 40, C40 tetraethers. So essentially what happens, you have 1, 2 on one side, 3, 4 on the other side. So 2 fatty acid chains from each side is covalently joined, and this is what we call as an archaeal monolayer. So this, think about that. This can be formed. This is just a monolayer which is found in the archaea, which is completely absent in the eukaryotes or in the bacterial structure. But these normal type of bilayer, phospholipid bilayers, are also found in many archaea as well. So let's take a look at these membrane lipids in a little bit detail. So what we mean by the structural differences between the phospholipid, which is different from the bacteria. So here what we are looking at, if you look at the phosphate and the glycerol linkage, and this is in a left-handed, the chirality is different in between the archaeal and the phospholipid of the bacteria and the eukarya. So here you can see the phosphate group and the glycerol. This is in the L isomeric form, and see that you change it, you flip it on the bottom from the top, and now this is in the D isomeric form. And this D isomeric form is a different chirality from this one, and this is actually seen from the bacteria and the eukarya. And the next one is, like, you can see that here the bonding is through the ether linkage, whereas the bonding with the same glycerol group is via the ester linkage. And here is your fatty acid tails, whereas see that the hydrocarbon is made of these isoprene unit, which also can have side branching, which is known as these phytanyl side chains are present. And we talked about this also, that you can have some of these diethers, which can be 20 carbon long. So when it's 20 carbon long, each is about 5 isoprene unit. So 4 isoprene unit, 4 multiplied by 5, can form these C20 diethers. And sometimes a C20 diether from one side can be covalently joined with a C20 diether from the other side to form a C40, 1, 2, 3, and 4, C40 carbon monolayer. So here you have a monolayer membrane. The outer leaflet and the inner leaflet are covalently joined, whereas here you have a bilayer where the inner and the outer leaflet are not joined, which is similar to the plasma membrane structure that we find in the bacteria and the eukarya. So these are some of the consideration of how the archaeal membranes are different. So here we already talked about that the archaeal membrane lipids, just as a recap, is glycerol diether lipids, hydrocarbon of 20 carbons attached to glycerol. C20 diethers make a bilayer, where the diglycerol tetraether each side you have one glycerol, so you have diglycerol, and two hydrocarbons of 40 carbons attached to two glycerol. And these tetraethers are more rigid than the diethers, and it forms a monolayer. So these polar phospholipid, sulfolipid, glycolipid, and many types of unique lipid modifications are also found in the archaeal membranes, which are also common in the bacteria as well as in eukarya. So here is another idea about the bacterial lipids, where you can see that these are the phosphate groups at the very top, and these are the glycerol with the ester linkage, whereas here you can see the archaeal lipids with the ether linkage, and which some of them may have these isoprene units and may have side chains. And here you can see that right here, this is essentially a monolayer. So this is a bilayer, but still it is an archaeal lipid. The facets of the bilayer is separate, whereas here the two leaflets of the plasma membranes are bonded to each other in here, forming a monolayer. So these are some of the differences that archaea have the branch chain hydrocarbons attached to glycerol by the ether linkages. So other than that, so outside the plasma membrane, so here we are looking at the plasma membrane. There are some types, I'm just pointing out to this number 5, where you can have a double plasma membrane. A plasma membrane encloses a small pace and another plasma membrane. So you can just think about that. If you have a single plasma membrane, we call this as the monoderm, whereas you can see that right here, this is essentially a diderm, or there are two membranes on top of each other. So usually the most common thing that what you see is the presence of the S layers. So these pink spherical structures, these are essentially the S layers. Sometimes there are some proteins which are also associated with some of these S layers that we also see in these areas. And there are some specific type of proteins that are present, and say for example, there is an archaea called methanospirillum, and there is essentially a moiety of methanochondroitin, which is a glycolipid that is present. So this chondroitin and the chondroitin sulfate is essentially found in association in the tissues of the eukaryotes, but this methanochondroitin is found in some. On the other hand, there could be some of the proteins which could be below the S layer. Sometimes there could be carbohydrate below the S layer, or in some cases, the S layer may not be present, and there could be proteins or carbohydrates in here. So these are all the different types that is essentially found. Also, one of the important thing is that the plasma membrane sometimes, just like the eukaryotes, may have some microdomains which actually concentrate some of these enzymes or proteins for its function. And one of them is found in one of the bacteria called Pyrococcus, and a protein called flotilin. They can essentially compartmentalize them in some of the microdomains on the plasma membrane that is also found. Now, interestingly, what we are going to see that a typical structure of the S layer can be up to 70 nanometer thick. And you can see that this is the hydrophilic membrane surface. This is from the top. And these S layer can be tethered with the plasma membrane and resembles a protein canopy and from the side, or a geometric pattern from the top. So essentially, the S layer can be decorated with a carbohydrate for the stabilization. But sometimes in between the S layer and the plasma membrane, the pseudomurine can be present in some of the archaea. So what is a pseudomurine? Pseudomurine is a similar structure to the peptidoglycan, which is also known as murine. That's why it is pseudomurine is false murine. So peptidoglycan is made up of N-acetylglucosamine and N-acetylmuramic acid. And they are joined together by a beta-1,4 glycosidic bond. And each one of them do have a side chain or stem peptide chain. And there are some unusual lipids, unusual amino acids. And these amino acids are sometimes with the D-chirality. So they are D-handed. On the other hand, what we actually see, particularly in the archaea, that instead of these N-acetyl acid, you have N-acetyl glucosamine, but along with that, you have a different acid which is known as N-acetyl acid. This minuronic acid is different from the muramic acid, and they are joined by a beta 1,3 glycosidic bond. So again, in the regular murine or peptidoglycan, N-acetyl glucosamine, NAG, is joined with N-acetylmuramic acid or NAM by a beta 1,4 glycosidic bond, whereas in the pseudomurine in the archaea, the N-acetylglucosamine is joined with an N-acetylminuronic acid with a beta 1,3 glycosidic bond. And also their side chain do contain the L amino acids, which are present in the eukarya. So they do not have D amino acid, but they are present in the L amino acid, and they're structurally, they are very different. And very interestingly, that the lysozymes, which can digest the peptidoglycan, or the penicillin, which can interfere with the synthesis of these interbridges that we have seen made up of glycine in between these stem peptides, these are ineffective in the archaea. They do not inhibit the archaeal enzymes which synthesize these interbridges or similar structures. And the reason is that they do not have to, because as we have said, that the archaeas are usually not usually, they are non-pathogenic. They do not infect human beings, and we do not have a threat from them. On the other hand, the archaeal extracellular vesicles and nanotubes are also found, and these are composed of the plasma membrane surrounding the cell wall material, or simply the S layer. Sometimes the cargo, which is a cytoplasmic, contain proteins, nucleic acids. These can be essentially packed into them. And essentially, these are thought to be important for the intracellular gene transfer and in thermophiles to protect the DNA from denaturing in the high temperature. So somehow, if the water temperature or something at one point of time actually increases, they actually form these tubes, and these float away from these high temperatures into areas where the denaturation may not happen. So these are some of the structures that you can see that right here, some of the EV in a high resolution in a scanning electron micrograph, and you can see in a transmission electron micrograph, you can see this extracellular vesicle formation, essentially these structures. And we do not really know much about these archaeal extracellular vesicles and nanotubes that you are seeing right here. These are the nanotubes, and we are just learning about these structures. So on the other hand, the archaeal cells use many of the same mechanism for the nutrient uptake exhibited in bacteria. And for that reason, as we have said, that there are some of the microdomains which can actually help in the passive as well, facilitated diffusion, active transport like a primary secondary transport. And along with that, there are phosphoenol yruvate sugar transfer system, which uses the group translocation mechanism that has been found in some of the archaea, known as PTS as well. So these are the overview of the cell envelope essentially. So after the cell envelope, let's take a look at that what is in the cytoplasm and contrast the cytoplasm with the bacterial and the archaeal cell. Very similar, they lack the membrane enclosed organelles and may contain the inclusion bodies, which contain many of these carbon sources or sometimes sulfur sources, iron sources, and contain gas vacuoles for the buoyancy. And along with that, you have other things like ribosomes, essential for the protein synthesis. You have a nucleoid region where you have the DNA, and also the plasmid, which are the extra chromosomal fragments, circular fragments of the genetic material. The cytoplasm do also contain some of these cytoskeletal proteins, and these are similar or homologous to the tubulin and the actin, and some of these are called the FTS-Z, SET-Z, or some of them is called also known as R-tubulin. And these are FTSZ, ZZ, R-tubulin are similar to the tubulin structures. And there are some of the crenactin and lokiactin, and these structures are similar to the actin, which also keep the shape of the archaea, and some of the structures could be different as well. So let's take a look at the ribosomes first. The same size ribosome, they are 70s, and they have the same 50s large subunit and the 30s smaller subunit, but they are very different from the bacterial ribosomes. The ribosomal RNA molecules is similar to the size of bacteria. The nucleotide sequence is different. So the small subunit do contain that 16S small subunit ribosomal RNA, but depending upon the sequence, which is very different from the bacteria, the archaea by itself is an entirely different domain. And also the protein composition actually differs. The archaeal ribosomes have more ribosomal proteins, and this is also another thing, that the different composition makes the archaeal ribosome unaffected by the antibiotics that target the ribosomes. So there are many antibiotics which have been developed or isolated from different microorganisms like a bacteria, which act on the 30th subunit or the 50th subunit or the ribosome on its own. And we are going to talk about that in the antibiotics chapter. And many of these antibiotics are completely ineffective in inhibiting the function of the ribosome for the archaea. It's not that it is needed for any disease purposes, but that just tells us that the proteins are very different, and that's the reason the target proteins which are affected by the antibiotics towards the bacterial ribosomes do not work essentially for the archaeal ribosomes. And the archaea is in that way more similar to the eukaryotic ribosomes compared to the bacteria. So the nucleoid region contains the circular chromosome and the nuclear associated proteins as well. And these nuclear associated proteins essentially do have some of these essentially compacting proteins. One of them is essentially known as the coalescin, which essentially compacts the DNA as such. One of the interesting part is that usually these are not and the other characteristics are very similar. It's an irregularly shaped region in the bacteria and the archaea, not membrane bound, with probably a few exceptions, and the evidence for polyploidy in archaea as well. So they may have duplicates of their genomes, so they could be polyploid. And as I said, the supercoiling are essentially aided by these nucleoid associated proteins, or the NAPs, and one of them, as I said, is coalescin. Along with that, there is some proteins which are present, which are very, very different from the bacterial protein, are the histones. But these histones, although named as archaeal histones, are very dissimilar compared to the eukaryotic histones. First of all, their sequences are very different. On the other hand, their organization is very different as well, and the nucleosome structures, which are related to those of eukaryotes, are very different as well. So in eukaryotes, the histone proteins, many different types, they forms octamers or structures of 8s. Around these 8 histone proteins or octamers, the DNA is enwrapped. Whereas what we essentially see, the histones or the archaeal histones are in a group of 4 or tetramers. But there are some of the exception as well. In some of the archaeal species, we find 2 histone proteins around which the DNA is enwrapped. We find as many as 12 histone proteins around which the DNA is enwrapped. But the typical structure is a tetramer histones enwrapping the the DNA compared to the octamer histones found in the eukaryotes. That's the main difference between those. But these histone structures for enwrapping the DNA and condensing the DNA is very, very different from the bacteria. And one of the idea is that as the archaea live in an extreme environment, this compaction of the DNA and the protection of the DNA may be provided by some of these histone proteins and associated proteins. So after that, we are looking at some of the external structures which are used for the attachment and the motility. And the first one is essentially the structure which is the pili. And the pili is essentially composed of the pilin proteins, and they are composed of, they are made in the cytoplasm and then anchored to a protein complex in the plasma membrane, usually in the S layer. And essentially what we have that there is a kind of a pili which is also known as an adhesive archaeal pili. And there are some of the pili which can be even UV induced. So these are known as UPS or UV inducible pili. And when these pili actually form, many of the archaea actually aggregate, and this helps in the DNA transfer. This is probably a survival mechanism by some of these pili. And this is essentially found in a particular species, which is known as the Saccharolobus species. In the Saccharolobus, this type of archaeal adhesive pili and UV-inducible pili is observed. Other than that, there are very specific type or structural type of pili. One is known as a cannula and cannulae plural. These are the hollow tube-like structure of the surface of many thermophilic archaea. And these are shown that the daughter cells arise from the cell division, remain connected to each other by this cannulae-like structure. And the hemi, on the other hand, resemble this grappling hook-like structure. And these are essentially for attaching the cells. And these are seen in some of the biofilm communities which associate with bacterial communities. So this is an association of the archaea and bacteria, and hemi essentially use the cells use the hemi to attach to the other structures. So here you are looking at some of these structures. See that right here is a hook-like structure that is essentially present in here. So these are some of the cannulae and hemi-like structures that is present in the archaea. So on the other hand, this archaea does have a very specific type of motility function. So the structure of the flagella and the archaea is actually much different. And the flagella are thinner than the bacteria, which is the archaea is essentially thinner. And they are made up of more than one type of protein, and this filament is not hollow. Remember that the bacterial flagella, the filament is hollow, and the flagellin proteins, they are essentially supplied from the bottom, and they go all the way up, and they are added at the top with this filament capping protein. They essentially grow from the top. Whereas here, there are very different structures. So these proteins, which is known as these ARC-B proteins, they are essentially synthesized, and they are associated with the S layer. And with the help of this ARC-J, they are added at the bottom. And the ARC-F protein essentially anchor the flagella on top of the plasma membrane along with the S layer. So ARC-F anchoring, and ARC-B is essentially the protein which essentially make the filament. With the help of the ARC-J protein, they are added at the bottom. And here you can see that there are several different types of protein, which is ARC-C, D, E, H, and these essentially is the motor complex and resemble the basal body. And out of those, this ARC-J is an ATPase. So a basic, very fundamental difference is also, other than being a solid flagella and the subunits added from the bottom, that the power for the archaea movement is the ATP hydrolysis, which is distinctly different from the bacterial flagellar movement, which is driven by the proton motive force or the PMF. And usually these comparisons are very important for your quizzes and the exam. So remember that the flagella is always powered by the proton motive force, which is the movement of the H plus ion gradient from the periplasmic space into the cytoplasm, whereas this ARG-J is an ATPase, which uses the power of hydrolysis of ATP to move these filaments. And the direction moves the cells forward, backward, rather than runs and tumbles. And swimming and motility at very fast speed has been observed as well. And essentially, as a take-home message, that we are still learning the fundamental basic of the structures, and you can see that compared to the wealth of knowledge that we have the bacteria, our knowledge is still limited about the archaea. So it is critical here to understand and remember the similarities and the differences between the archaea and the domain bacteria.